In the last unit, you had a chance to see how variation could get passed on. But what's one of the mechanisms that causes variation? Well, that's mutations. And so we're going to look at two different types of mutations, and the first one we'll start with are point. Point mutations can often be caused by some sort of outside energy source. It could be an incoming photon, which would be like energy from the sun or radiation. It could come from carcinogens sometimes, or different other environmental chemicals. But what they can do is break apart the DNA strand and affect the actual base pairs that are in DNA. A couple different things. So if we start here and we look at our normal strand of DNA there, you'll notice the pattern GUUC, GAUU, GA. And in a substitution mutation, one base pair here gets swapped out and another one is put in. So instead of having the A and T like you have here, it appears we now have a C and a G. And remember, that can affect the different codons that are going on there. Another type is insertion. An entire DNA sequence gets inserted there. In this case, it happens to be a triplet, which just means there'd be another polypeptide or peptide that would be in there, amino acid. But it could be, if it's just one base in there, it shifts everything and changes all of the amino acids in the sequence after there, and even affects the stop codon that would tell it when to stop. Likewise, if there's a deletion, in this case one base pair is deleted, it can cause a frame shift, which goes across and once again can not only affect every amino acid after that, if it's one or two and not a multiple of three base pairs, but also the stop codon at the end of the DNA sequence as well. So here's a couple examples of how this might look in words. You can see we've got these three codon sequences, and it would be translated as the old man and the C. In a substitution, one letter is changed, and now it's the old man and the T, which is an entirely different meaning. Another thing that can happen is you could get a deletion here, and in this case, one of the N's was taken out. The and old are fine, but everything after that changes and is affected and messed up. The last one, we've got an insertion here, which is the X, and now the first word is fine, but everything else after it changes. And once again, if you've got some stop codon that's supposed to be here, it's not going to be the same anymore, and so that protein just won't form properly. Now, do all mutations make a difference? Well, look at the following two sequences. We've got a point mutation here, which is a substitution. Once again, those point mutations aren't going to be nearly as bad as a frameshift mutation because it'll only change one of the amino acids sometimes. And if you look here and you look at our chart that we've used before, what you'll notice is you still have the start codon, you still have the serine and the stop because of the other ones, but both of these, as you look at the chain here, both of these right here code for the same amino acid, so that point mutation will actually have no effect on the gene or the DNA sequence. So if you were looking in order of importance, frameshift mutations typically will totally affect the gene being produced. And remember, 99% of those changes are probably going to be harmful, but every once in a while it might create a new protein that can actually be helpful. Then substitutions tend to be the least effective because they might only replace one amino acid in the sequence. However, remember, if one amino acid gets switched, that can still change the structures of the protein, especially like the tertiary and quaternary structure, how it folds upon itself. And then the least would be this point mutation where you still have the same amino acid at the end. So those are two main types of mutations: point and frameshift. Our substitution is a point mutation, and deletion and insertion are frameshift mutations.